Hello, everyone. Good morning, good afternoon, good evening. This is Dr. Kaz. I'm doing another one of my infamous videos today. Today, we are going to be looking at Chapter 2, Chemistry or Biochemistry, and we are going to be picking up and talking about one of our classes of macromolecules. We've previously covered lipids, and now we're going to continue on with carbohydrates, then proteins, then nucleic acids. So let's get down to it. Carbohydrates, these are the macromolecules that are commonly referred to as sugars. But pretty much what you're going to see in our carbohydrate class when you're looking at the molecular formula, you're going to see three types of molecules: carbon, hydrogen, and oxygen. And essentially, each carbon is going to have its own hydrogen attached to it and its own hydroxide ion, which is OH, attached to it. And so when we're looking at carbohydrates, there's a couple different kinds that we're going to be seeing. We're going to see our monosaccharides, mono being one, disaccharides, di being two, polysaccharides being many. So monosaccharides are commonly referred to as simple sugars. Disaccharides is when we take two simple sugars, attach them to one another, and then we form that molecule. And then our polysaccharides, when we essentially take several monomers or monosaccharides and just keep linking them to each other to where we have many or several monosaccharides attached to one another, and that is our polysaccharide. We're going to kind of move through that. As you move through biology in this course, you're going to find out that glucose is a very, very popular and important molecule. It is a carbon carbohydrate, most common monosaccharide, and in fact, when you're looking at the disaccharides, it is always going to be one of the two possible monosaccharides involved in a disaccharide. More on that in a few moments. Glucose is considered to be the primary nutrient when we're talking about energy for cells. In fact, it is the preferred nutrient. And so you'll see in this chapter and then in future chapter, when you take 211, Bio 211, we're going to talk about the importance of glucose. But because glucose is such a crucial and important molecule, our body needs to carefully maintain the levels inside, especially in our blood, also in our cells. So we'll talk more about that. So since glucose is so important, your body has come up with an important means of kind of saving glucose. And that important means is the creation of this molecule called glycogen. So glycogen is a polysaccharide. So several monomers of glucose molecules are stacked and linked to one another. And you're going to find this molecule, and this is important to know, in your liver and in your skeletal muscles. So think of glycogen as just the stored form of glucose. And the process of making glycogen in one of these two tissues is called glycogenesis. Glyco meaning meaning glycogen, genesis meaning the creation or the making of. So glycogenesis is the making of glycogen in the liver and skeletal muscle. Now, when there's a situation in which you need energy and there's not enough of that glucose floating through your blood, then we need to start to break down glycogen into its monomers, glucose. So the liver is going to undergo a chemical process called hydrolysis, and it will start to cleave off glucose monomers off of the glycogen molecule. This process is called glycogenolysis. So again, there's that word glycogen in the title of this process, and then lysis following the name. And so lysis means to break. So glycogenolysis is the breaking down of glycogen into individual glucose monomers there. The liver is also very adept at making glucose from what we call non-carbohydrate sources. That would be an example of an amino acid, which is a building block for protein. We can utilize, right, different non-carbohydrate sources, fats, lipids that we discussed before, and we have this process called gluconeogenesis, again, performed in the liver. Gluco meaning glucose, neo meaning new, genesis meaning to make. So gluconeogenesis is this process that the liver is going to perform in order to create glucose from our non-carbohydrate sources. So this slide here, you can kind of see, right? You can see our glucose monomer on the left, and then our glycogen molecule on the right. And so if you follow the arrow here at the top of our page here, that's going from left to right, you'll see when we start to attach glucose monomers to one another, we create the glycogen polysaccharide molecule through the process of glycogenesis. And then the opposite happens when we go from the right side of our screen to the left side of our screen, following the arrow at the bottom, called glycogenolysis, excuse me, I can't talk today. And that's essentially where we just start to cleave off these individual monomers here to give us our glucose molecule, which will then be dumped into the blood, and then it'll be transported throughout to the body to the tissues and the cells that require glucose for whatever their metabolic needs may be. Okay, so let's talk about some of the other types of carbohydrates that we'll see in the body. We have a couple other simple sugars or monosaccharides, and those are hexose monosaccharides, which means a six carbon sugar. And so if you recall earlier in this chapter, we talked about isomers. These are molecules that have the same number of atoms. And in this case, glucose is going to have some isomers called galactose and fructose, and these will be the other monosaccharides. And some other monosaccharides that we will talk about later on are our 5-carbon monosaccharides, and those are called pentose sugars. Pent being 5, so pentose sugars. This will include ribose and deoxyribose. Those names might be slightly familiar to you because when we're talking about DNA and RNA, there's a sugar molecule involved in those structures, and so those will be the sugar molecules, those pentose sugars. More on that shortly. Our disaccharides, we talked about this earlier, are going to be our 2 monosaccharides that are bonded together. Now, keep in mind, I said previously that all the disaccharides are going to have at least one glucose molecule in that structure. So when we talk about our disaccharides, we have our table sugar sucrose, right, our milk sugar lactose, and then our malt sugar maltose. So in all 3 of those molecules, one of the monosaccharides is going to be glucose. And then the other one will be either galactose, fructose, or another glucose bonded to a glucose. All right, so I'll show you a picture here on this page. At the top, you can see the monosaccharides. And so there you see on the left side of the screen, galactose and fructose, and those are our 6-carbon sugars. And then on the right side of the screen, you see our pentose sugars, the ribose and deoxyribose. More on that shortly. So now what we're going to do is we're going to take a glucose molecule, shown here in green, and we're going to bind it to another monosaccharide here on the left side of our screen. So if you look here on the bottom part of your screen, sucrose is made up of a glucose molecule and a fructose molecule. The next sugar is lactose, our milk sugar. Again, we have glucose, but in this case, it's bonded to galactose. And then finally, on the right side of our screen at the bottom, you have maltose, and that's essentially just 2 glucose monomers or monosaccharides there. So that's what we're looking at here when we're talking about our disaccharides. So keep in mind, just to reiterate, disaccharide always has at least one glucose molecule or monosaccharide that's bound to another monosaccharide. All right, Continuing on with some of the other types of carbohydrates, we talked about monosaccharides, we've talked about disaccharides, and now we're going to talk about our polysaccharides, poly being many. So in this case, we'll have 3 or more sugars that are going to be present in these molecules. So when we talk about the polysaccharides in animals, that's our glycogen. So animals will take those monosaccharide glucose molecules, attach them to one another, make this big long chain, and they'll produce glycogen, which we talked about, which is stored in the liver and the skeletal muscles. In plants, we refer to polysaccharides as starch and cellulose. So starch is great, all right, when we're using it for dietary nutritional means, all right, because it's great for us to get glucose from during the digestive process. Cellulose is also a good plant polysaccharide in the fact that we refer to it as fiber because it is non-digestible. So you might be asking yourself, why is that a good thing if it's non-digestible? Well, sometimes we need what they call quote unquote roughage, something to help facilitate the digestive process. And cellulose is that substance, that fiber, that non-digestible material that we take in, all right, during the digestion process that helps facilitate and manage digestion a little bit more. When you get into the digestive chapter, you'll learn more about that. Another type of carbohydrate are our glycosaminoglycans. You may have seen this before, all right, known as GAGs. These are large carbohydrates with an amine group, and that's a nitrogen-containing group. And these molecules were seen in the ground substance, all right, of our connective tissue. And so these GAGs, these glycosaminoglycans, all right, when they're attached to a protein, we call those proteoglycans. And you'll see the glycosaminoglycans and the proteoglycans all throughout the ground substance of connective tissue. And so you probably don't know what that means, but that's okay. When you get to chapter 5 on tissues, you'll learn all about the importance of these glycosaminoglycans and these proteoglycans, how these large molecules play a role, all right, with water and whatnot, but more on that in chapter 5. So, question, what is the repeating monomer of glycogen? Give yourself a second. I'll repeat the question. What is the repeating monomer of glycogen? If you said glucose, you would be correct. So where is glycogen stored in the body? Give yourself a second. One more time. Where is glycogen stored in the body? 2 places. Stored in the liver and in the skeletal muscles. So if you answered those both correctly, good. You're starting to understand the material. If not, do not worry. Watch the video again, and it will help you get that answer. So let's move on to our second, or the actual third macromolecule, but the second that I'm talking about in this video, and that is proteins. One of my favorite macromolecules. And if you look at this long list of protein functions, you'll pretty much see that proteins are involved in almost everything that happens when we're talking about the physiological processes of your body. Synthesis, digestion, support, movement, transport, membrane transport, protection, defense, storage, regulation. And as we go through our course of Bio 210 and Bio 211, you are going to see all of these protein functions, right, on display. Digestion, right? Enzymes, and we'll learn what enzymes are, but they are protein catalysts. Much like a lighter is to making a fire, enzymes are to a chemical reaction. We will talk about in chapter 4 the cytoskeleton. We have our normal bony skeleton that helps to give us support to our bodies. So you'll see in chapter 4 that our cells have their own version of a skeleton, which we call the cytoskeleton, and proteins play a large role in that structural support. When we get into the chapter on muscles, we're going to learn about 2 very important proteins called actin and myosin. They're involved in our sarcomeres for our cross-bridging mechanism. And so they aid in body movement during the contraction and relaxation of muscles. In 211, you'll learn about a very important protein molecule called hemoglobin, which is this protein molecule that will bind our respiratory gases in our blood, and one of which, those respiratory gases, is oxygen. Oxygen is crucial for cellular aerobic respiration. Then we have membrane transport. Again, you are going to learn in the next chapter, in chapter 4, right, these specialized carrier proteins that are going to be found inside the plasma membrane of your cell and what role they play in the movement or transporting of substances into the cell and out of the cell. In 211, you're going to see, right, how proteins play a role in defense and protection when we talk about the immune response, specifically that of the immunoglobulins, which are our antibodies. A lot of what you may have heard during the whole COVID-19, right, epidemic. We'll discuss that in more detail when we get to that chapter. And then finally, we have our storage and regulation. And again, as we move through this course, you are going to see how proteins are involved in storage and in how they are involved in overall just regulation for certain processes and physiological reactions in the body. So let's take a peek here and look at the general protein structure. So essentially, proteins will be made up of the building blocks that we call amino acids. So amino acid monomers are going to be attached to one another. And then you'll essentially, once you reach a certain number of amino acid monomers, then we will actually create a protein. So we have about 20 different amino acids that are to choose from to make our protein structure. But what we will see on these amino acids, on one end of the amino acid, you will see an amine group, which is going to be a molecular group that contains at least a nitrogen. And then you're going to see on the other end a carboxyl group from carboxylic acid, and that is going to be on the other end of our structure. And so normally when we're talking about amino acid, you'll have a carbon molecule, and then on one side of it, so we're looking at it here in a moment on the following slide, you'll see to the left of the carbon, you'll see an amine group. To the right of the carbon, you'll see a carboxyl group or a carboxylic acid group. And then normally you'll see on that same carbon, because carbon can form 4 covalent bonds, so it forms one covalent bond with the amine group, one covalent bond with the carboxyl group, then it'll have another covalent bond with the hydrogen, and then another covalent bond to complete its 4th covalent bond with what we call an R group. And these are a side chain structure. You'll see what I mean when I show you a picture here. So quickly, I'm just going to show you what I just was talking about. We'll zoom in here so you have a better view. So here's our amino acid. So you have the carbon group. Here's the amine group. There's our nitrogen. So we know that's the amine group on the left. And then on the right of that carbon, we can see the carboxylic acid group here to the right. So carboxyl group, an amine group. Here's the hydrogen atom attached to the carbon, and then we have our R group, which is what we call the side chain, and then you have a variety of different structures that can be attached to the carbon. So you see 1, 2, 3, 4 covalent bonds that carbon can form. So keep that in mind as we're kind of going through this. So now we're going to talk about how we're going to eventually create a protein molecule. So it all starts with just starting to link these amino acids to one another. And so to do that, we're going to start to covalently bond or covalently link amino acids to one another through what we call peptide bonds. And so in order for a peptide bond to occur, right, we're going to see a loss of something from the 2 amino acids that we're going to bond together. So one of the amino acids is going to lose one of its hydrogen molecules. So for example, this hydrogen molecule or the other one could be dropped from this molecule here. And then the other molecule or the other amino acid will lose its hydroxyl or OH group from the carboxylic acid end. And if you notice something about both of those, you see an H, you see an OH. You add that all up, that's H2O. So when two amino acids are covalently bonded to one another to create a peptide bond, this is called dehydration synthesis. Right? So when you think of dehydration, you should be thinking of water. Right? So someone says, hey, you're dehydrated. You're thinking of loss of water. Well, that's exactly what happens. So when these two amino acids are covalently bonded with one another, we actually get water from this reaction here. So now that we have a peptide, when we bond both of these amino acids together, you're going to see at one end of this new molecule, we're going to have an amine group, and that's what we call the N-terminal. And at the other end of this molecule, we're going to have our carboxyl group, right? That's our carboxylic acid, and that's going to be our C-terminal. So again, let's show it in picture form, make it easier to understand. Here's what happens. We have our amino acids. So we're going to take two amino acids, so two molecules that look similar to this, and we're going to attach them to one another. Here's one amino acid, here's the other amino acid. So the carboxylic group loses its OH structure here, and then our amine group on the other amino acid loses its hydrogen structure. And so H2O, as a result, the formation of a peptide bond, we will get water from this reaction. And so when we put these two individual amino acids together, we now form what's called a dipeptide, meaning two peptides. So what this picture is showing you at the bottom is a polymer chain or a protein. And so when we start stringing amino acids with one another together, you have the beginning of the molecule, or I shouldn't say the beginning, but you'll have one end of the molecule here on the left side of our screen and the other end of the molecule here on the right side of the screen. You'll notice one end has the amine group, there's the nitrogen, we call that the N-terminal. The other end has the carboxylic acid group on it, and that's the C-terminal. So that's what I was referring to when I was talking about the N-terminal and the C-terminal. Okay, so now we're starting to see how we're building these structures. So when we start to just stack these amino acids, right, onto each other, we give them certain names depending on the number of amino acids in the strand. So if we have anywhere between, we saw what happens when we have two amino acids, we call that a dipeptide. So when we have 3 to 20 amino acids linked together, we call that an oligopeptide. If we have 21 to 199 amino acids linked together, we have a polypeptide. And once we get over 200, so more than 200 amino acids, then we finally have our protein. All right, so just kind of be familiar with these terms. You're going to hear these terms, oligopeptide, polypeptide, protein, especially when you get into the digestion chapter and you're going to learn about how we break down proteins, all right, protein catabolism, right? So just kind of be familiar with these. I'm not going to be specific on the number of amino acids, but you should have a good idea, right? It's a good idea to memorize that. All right, glycoproteins, again, glyco meaning carbohydrate. So a glycoprotein is going to be a protein with a carbohydrate group attached to it. This becomes very, very important when we talk about blood typing. So you'll have these certain protein structures, these glycoprotein structures on the surface of your red blood cells, and they're going to determine what we call your ABO blood typing, whether you're A blood group or B blood group or O or AB. So glycoproteins play a big role in that. All right, question time. First question, what are the monomers of proteins? Just got done talking about that. Let me repeat that one more time. What are the monomers of proteins? If you answered amino acids, then you are correct. And what attaches one amino acid to the next is going to be that peptide bond that yields protein. Excuse me, that yields water, right? That dehydration synthesis reaction. All right, second question. What is the name of the bond between them? And I just said what that bond was, right? Peptide bonds. All righty. moving on with our amino acids, right, and proteins, we're now going to quickly talk about how we organize those groups that I was mentioning to you that come off of that central carbon there, right? What we call the R group. So we have non-polar amino acids, we've got polar amino acids, we've got charged amino acids, and we have amino acids with special functions. So, charged amino acids, right? These R groups, real simple, are either going to have a negative or a positive charge. That's it. All right. So therefore, these amino acids, right, these ionic bonds that are formed, all right, between these amino acids will have a negative or positively charged ionic bond there. That confers that these amino acids are hydrophilic, meaning water loving. So they can interact in a water solution, no problem. Right? Amino acids with special functions, for example, we talk about one of the amino acids is proline. And so you saw in the previous slide how we just started linking amino acids to one another and they made this long chain. Well, protein can put a bend in that chain. Cysteine can form what we call a disulfide bond. And so when you dissect that word down, di meaning two, so you can have, right, two sulfur molecules bonded to one another. And then methionine, which is a very important amino acid because it's the first amino acid, no matter what type of protein you're making, no matter what. It's always, always, always the first amino acid during protein synthesis. So if you go back, or not go back, but if you decide to go through and read chapter 3, we don't cover it in this course, but chapter 3 talks a lot about the transcription process, the translation process, and it does mention methionine. So just keep that in mind. Throughout this course, and when we talk about certain chapters and certain physiological reactions, you're going to hear me mention the term denaturation. And so denaturation is this process which we'll start to see what we call a conformational change to a protein. And what that essentially means is these proteins that when they're being made, eventually once you get over that 200 amino acid mark and you get to the desired number of amino acids to make a specific protein, what you can see is there's different shapes that proteins can take on. It's almost like an origami type of paper folding situation here. And so what will happen is, depending on the protein type, it can be folded into a certain shape or configuration. And then once that occurs, then that protein, right, once it's been fully modified and the process is complete, then that protein can become functional. Well, with denaturation, we're essentially going to kind of unfold that protein. And when that starts to happen, we start to interfere with the activity or the function of the protein. And essentially, we can make a once functional protein non-functional. Unfortunately, this is irreversible for the most part. And we will see this in situations where you have a change in temperature, or, and you'll see me talking about this quite a bit, when we see a change in the pH. And so a lot of times I'll mention how when the pH starts to drop, when a solution starts to become more acidic, I talk about the risk of denaturation. That goes in the other direction too, meaning if the pH increases, if the solution becomes more basic or alkaline, you can also get denaturation. Point being is, if that's why it's really important that you have your pH values in your blood between 7.35 and 7.45. When you stop to drop outside of those ranges there, then denaturation becomes a risk, and you can start to see the unfolding of these proteins and then cease their activity and their function. So this slide here shows you just what we talked about, just some of those different amino acids for those classes that I was referring to, the nonpolar, polar, the charged, and then our special function molecules there. Again, just take a glance at them so you're kind of familiar with these amino acids. I'm not going to ask you to know the molecular formula or tell me if it's a charged amino acids or if it's a nonpolar. Okay. Alrighty, folks, the last of the macromolecules that we are going to talk about are going to be the nucleic acids. So our nucleic acids are going to be a macromolecule that has a couple functions: storage and transfer of genetic information. It's pretty important, all right? Pretty important, especially when we talk about making proteins and whatnot. So we have two classes of nucleic acid. One is deoxyribonucleic acid, otherwise known as DNA, and then ribonucleic acid, otherwise known as RNA. And so we're going to talk about the structures and what we're going to find, but essentially, right, we are going to refer to these molecules as polymers that are going to have what we call a nucleotide monomer involved. And then these monomers that are going to be attached to one another through covalent bonding, we're going to be using a phosphodiester bond here. So let me show you. Here are the three components that we're looking at. Right, so you'll have a sugar, a phosphate group, and a nitrogenous base. So when we talk about this nucleic acid, our nucleotide monomer, here's the sugar right here, right? And that is going to be a pentose sugar, 5 carbons. Then we have our phosphate group here. And then finally, we're going to have our nitrogenous base here. And so the nitrogenous base, you can see here, you have a double ring structure, but we can also have a single ring structure. So the nitrogenous base can vary when we're looking at our nucleotide monomer. So again, our nucleotide monomer is going to have 3 things. A sugar, that's our 5 carbon sugar. Also, we'll have our phosphate group that's going to be attached onto the sugar. And then finally, we have our nitrogenous base, which is going to be a single ring or a double ring structure that will also be attached onto the sugar. All right, so when we're looking at the nitrogenous bases, there are going to be 2 classes: pyrimidines and purines. Pyrimidines are going to have 1 single ring, purines have a double ring. How I remember which of the nitrogenous bases are going to be in the pyrimidine class, I just think of CUT, C-U-T. And if you look at a pyramid, it's a triangular shaped structure, and the tip of it always seems sharp to me, and you can cut your finger on it. That's how I remembered it. So when we're looking at the pyrimidines, the 3 molecules that you're going to see, the single ring bases, are going to be cytosine, uracil, and thymine. The purines, which are the double ring bases, the 2 bases that you're going to see here are going to be adenine and guanine. Adenine and guanine. So here you can see a picture of the nitrogenous bases. On the top, we have the pyrimidines, CUT. Right? And so you'll see here that some of these nitrogenous bases are going to be in both DNA and in RNA. Some will be in RNA only, and some will be in DNA only. So what I recommend that you do is, when you're trying to learn this, just learn the ones that are in RNA only and DNA only. There's only 2. And what I mean by that is uracil is in RNA only, thymine is in DNA only, and then the remaining bases are in both. To each their own, though. If you have a better way, but that's usually how I like to do it. So uracil is in RNA only, thymine is in DNA only, and then the rest of them you can find in both. So let's talk about DNA. Our double-stranded nucleic acid, deoxyribonucleic acid. Where do we find this? The majority of your DNA is going to be located in the chromosomes on your nucleus, but you also have some chromosomes in your mitochondria, and that's out in the cytoplasm, right? You do have a couple out there, and those, just FYI, those chromosomes, that DNA found in the mitochondria comes from your mommy, from mother. Right, it is maternal DNA. Easy to remember, mitochondria M, maternal M. Right, so mitochondrial maternal DNA, right, is found in the mitochondria. All right, so what are we looking at when we're seeing the DNA? Right, we're going to have our sugar group, so our deoxyribose sugar that is going to be attached to its phosphate group, and then one of the 4 nitrogenous bases that we find in DNA. Remember, we don't see uracil in DNA, okay? No uracil. Everything else, yes, no uracil. So when we look at the DNA molecule, it looks like a ladder that somebody just started to twist, right? So it's a double-stranded molecule, and those bases that we just saw right up here, these are the bases, adenine, guanine, cytosine, thymine. They'll be bonded to one another through hydrogen bonds. I'll show you a picture. In fact, let me show you now. So here you can see on one side, let me zoom in here. On one side of our double-stranded DNA molecule, you'll notice, right, we have the adenine base. On the other side, we have the thymine base. And so they're bonded to one another through these hydrogen bonds. Right? And I'll talk about this here in a second, which bonds with which, right? But keep in mind, on opposite sides, you'll have, all right, the bases make hydrogen bonds with one another. Right? So you have the rungs of the molecule, and then you have, right, the sides here, which is escaping me what we call those, right? But you'll notice that the rungs are going to be, right, the nitrogenous bases there. Right, so which binds with which? Well, I always think that G and C look somewhat similar to one another. So guanine is paired with cytosine, and then adenine and thymine are going to be paired with one another. And that's how it works in DNA. So you'll always see that pairing. Adenine will not bind with guanine, and thymine will not bind with cytosine or guanine, all right? It's just always going to be A with T and G with C. All right, ribonucleic acid, known as RNA, this is a single-stranded nucleic acid, so it does not look like a ladder. Right, and so where do we find this? Again, we will find this type of macromolecule in the nucleus and also out in the cytoplasm. Right, so we'll see both out in the cytoplasm, except for DNA. All right, when we say it's out in the cytoplasm, it's inside the mitochondria here. Right, so our ribonucleic acid, similar type of scenario. We're going to have its makeup being made up of ribose sugar that's bound to a phosphate group, and then one of 4 nitrogenous bases listed below. But keep in mind, no thymine in RNA. You got adenine, guanine, cytosine, and uracil, but no thymine. So here you can see on our picture, RNA is on the left, single-stranded, DNA is on the right, double-stranded. So there's a couple differences there. Obviously, this is a big difference here. You have double-stranded versus single-stranded. And then which nitrogenous bases will be found in which of those molecules there. And then finally, our last molecule that I want to discuss is ATP, a very, I'll say it one more time, very important molecule. All right, so ATP stands for adenosine triphosphate. TP is triphosphate. Similar type of situation that we just learned. You're going to have a sugar group, right? And so there's our ribose sugar that is going to be attached onto a phosphate. And in this case, it's going to be a 3-phosphate molecule. And then we'll have our nitrogenous base, and in this case, it's adenine. So if you look to the right there, you're going to see, all right, how this molecule looks. You've got the sugar group attached onto the nitrogenous base, and then you've got the phosphate group also attached onto the sugar. And so the importance of this molecule, and I'm going to circle it here, is that it is involved in the chemical transfer of energy inside the cell. I like to refer to it as the energy currency of the cell. And so when you look here at the molecule on the right side of the screen here, you'll see you have 3 phosphates. And these phosphates, especially the last 2 phosphates, right, are bonded to the other phosphate group through these energy-rich bonds. And that's essentially what will happen when we try to get energy from this molecule. We just snap these bonds here. So this last bond here has the most energy, and then the next one has a little bit less energy. So we can snap these bonds and release energy. And when that energy is released, we're able to utilize it to do work, to make something, to break something down. But regardless, all right, that's the importance of this molecule, that it contains those energy-rich phosphate bonds that we can break down or break and release energy to do some sort of activity. And so you'll learn later on that there are certain molecules in the body, like nicotinamide, adenine dinucleotide, or known as NADH, and then flavin adenine dinucleotide, FAD. And you're going to learn, we're going to utilize these nucleotide containing molecules in our electron transport train, in the Krebs cycle, or the citric acid cycle. That's the process of making ATP, right? When we have oxygen, right? We're going to see how we can use these molecules to help make ATP, but that's not going to be covered in this chapter or in this video. That's for another chapter and for another video. All right, folks, I hope you enjoyed this video. I hope you learned a lot. At any time, make sure if you don't understand something, pause it, rewind it, and see if you can watch it again to maybe pick something up that you didn't understand, or something that you missed. I hope you enjoyed the video, and I hope that you learned something. Have a great day. Please keep studying.